Today, scientists around the world are manipulating the source code of life itself, DNA. Though CRISPR gene editing is still in its infancy, some of the latest developments seem to be bringing us to the edge of a brave new world. One filled with designer babies, no diseases, bodies that never age, and a host of species with strange new features. CRISPR could, in the truest sense of the phrase, utterly transform life as we know it. The key to genetic modification can be found in deoxyribonucleic acid, or DNA. It guides the growth and development of all living things, acting a little like computer code. Our DNA contains 4 nucleotides, adenine, thymine, guanine, and cytosine, that are paired throughout the structure of the double helix molecule like rungs on a ladder. These rungs carry specific instructions, and the sequence of instructions ultimately makes up an organism's genes. Change the instructions, and you change the organism carrying it. Every cell in your body has around 3 billion base pairs of DNA code inside it. Just a couple errors in this code could create debilitating illness. Given the complexity of our DNA, it's a bit like misspelling one word in a stack of 1,000 Bibles. But CRISPR could act like a spell checker. CRISPR works thanks to a unique defense mechanism found in bacteria, which they use to defend against viruses. Some bacteria produce specific proteins that fight off viruses, and when this defense system succeeds, enzymes in the cell take parts of the virus's genetic code and store it. Like a patrol team with a mugshot, the enzymes carry around the virus's genetic code in the cells. If the virus with the matching mugshot attacks again, the cell is able to recognize and cuts the matching part of the virus, killing it. This cutting is very precise, which is precisely what makes CRISPR so exciting. Think of CRISPR-Cas9 as a DNA surgeon. It gives researchers the ability to cut DNA exactly where they want, adding or removing parts where needed. Moreover, it works on microorganisms, plants, animals, and humans. Genetic engineering using CRISPR is already weaving into our daily lives in some unexpected ways. Companies are using CRISPR to create allergy-free food, better biofuels, more nutritious fish, and decaf coffee beans. Scientists are also using the gene editing technique to eliminate muscular dystrophy in certain dog breeds, create faster racehorses, and control the numbers of animal species that transmit infectious diseases. We have even created glow-in-the-dark plants and animals. Yet CRISPR's true power lies in medicine. Researchers from Temple University have used CRISPR gene editing along with antiretroviral therapy to eliminate HIV infections in mice. In a few decades, CRISPR therapy techniques could also be used to disable a range of viruses that hide inside human cells, or even edit our immune cells to be better cancer hunters. It is very plausible that advanced gene editing techniques could be used to modify human embryos in the distant future. A simple procedure could help ensure your child is born immune to diseases. But why stop there? We could engineer humans with specific super traits that would allow them to survive in hostile environments on alien planets. However, we shouldn't get too carried away. CRISPR is still in its infancy. It will take decades to bring many of these use cases to life, and that's assuming that we actually can bring these use cases to life. Some experiments have shown that CRISPR causes inadvertent changes or off-target edits to DNA. Such changes could be harmful to life and override any positive effects, and other use cases could ultimately prove too complex to ever carry out successfully. Yet how many other things have we brought to life that were thought to be impossible in past decades?